So osmosis. Osmosis is the movement of water across a membrane. And we touched on this very briefly in the anatomy chapter, cell anatomy chapter. But before we talk about osmotic, I'm going to get into this a little bit more now because I know some people needed a review on it. So let me give a review of some vocabulary. A solution is a liquid that contains a solute, and a solute is a substance dissolved in liquid. So a solution could be like salt water. Salt water is a solution because it has a liquid, and that's the water, and it has the salt, and that's the solute that's dissolved in the liquid. Okay. If solute concentration is high, it means water concentration is low. Okay. Let's just say you have a glass of pure water and a glass of salt water. The water concentration, of course the salt concentration, we know where the salt concentration is highest, right? The salt concentration is highest in the salt water. The water concentration is highest in the glass of pure water. And the reason is, if those glasses are the same size, if they're the same volume, and in one of them you have nothing but water molecules, and in the other you have some mix of water molecules and salt water molecules, well, in the salt water that means there's not as many water molecules in the same space because some of the space is taken up by salt. I know it's hard to imagine that because we always imagine like the salt seems to disappear, so it must just fit in magically without changing the volume of the water, and that's just not true. So whatever you add to the water, it's going to change the volume even if it dissolves. So if you're mixing water with salt and it's the same volume of water as the pure water is going to be a higher concentration of water because it's nothing but water molecules in the same volume as opposed to the salt water, which has a mix of salt and water molecules, meaning that some of the space is taken up by salt and there's not as much space for the actual water. Okay, so the water concentration is lower where the salt concentration. So water concentration is low where solute concentration is high. Okay. Now, water moves down its concentration gradient. Water is going, just like every other molecule, water is going to move from high to low concentration. So that means water is going to move from pure water, where it's high concentration of water, to salt water or sugar water or whatever, wherever the solutes are, where water concentration is low. Okay? So you can think to yourself, water follows solute, but what's really happening is the same thing that happens with any other molecule. We're used to thinking about the molecules dissolved in water, but water is a molecule too. And all water molecules move to from where they are they're highly concentrated to where they have low concentration. Okay? So water moves from pure water to more solute-y water. All right, so what this does with animal cells, the way this matters for cells is that you got to think about the relationship between what's dissolved on the inside of the cell and what's dissolved on the outside of the cell to figure out where the water is going to move. Okay, so let's say you have a situation like this. You've got some cell floating in water, and these red dots, those are some solute. The solute could be salt, it could be water, it could be proteins that are dissolved in there. It doesn't matter what's dissolved in the cytosol. All that matters is there's stuff dissolved in there, right? So those red dots are taking up some of the space. The white space is the water. The red dots are the solute. Where is there more white space? Well, there's more white space outside, right? There's some red dots outside, but the white space is more concentrated outside and less concentrated inside. So the water concentration is higher. The water is more pure outside this cell. The water concentration is lower. The water is less pure inside the cell. Now, if you're thinking about the solute is the opposite. The solute is, of course, higher concentration inside than outside. So if the solute, the red dots, if they could move, they would move out, right, until they were equal concentration in and out. But the purpose of the cell membrane is to keep them in. So the cell membrane is not permeable to this solute, so the solute can't move. So what's going to happen instead? Well, instead the water's going to move in. The reason water can move in is because all cells have pores called aquaporins that allow water to pass. Okay, so there are channels that allow water through. So the solute can't move out. That means the water is going to move in. The water is going to move from where it is most highly concentrated to where its concentration is low. So it's going to move into the cell, and the cell is going to swell, and the water would keep moving in until there's equal concentration inside and outside. But what's actually going to happen here is that it's going to move in, and the cell membrane is going to swell, swell, swell. Before the inside is as dilute as the outside, the cell's going to burst. We call this cell lysis. Okay. This is osmotic lysis. Lyse just means to break. To lyse something means to break it. Osmotic lysis means the cell breaks because of osmotic pressure, because of water osmosing into the cell. Okay. So water follows solute, or water moves from where it is most pure to where it is less concentrated. So let's talk about this vocabulary, tonicity. You've seen these before. There's hypertonic, hypotonic, isotonic. All right, these are relative terms. Something can't be hypertonic by itself, right? Because that's like saying something is greater by itself. You have to say greater than what? Hypertonic to what? It can't be greater by itself. It has to be greater than, okay? So hypertonic to. So hypertonic means some solution has a higher concentration of solute, the dissolved stuff, than another solution. So salt water is hypertonic relative to fresh water because salt water has more solute than fresh water. And I remember that hyper means more than, more solute than. Hypertonic means more solute than, because if you think about a hyperactive kid, they have more energy than the average kid, right? More than. Hyperactive, more activity. So hyperactive, more activity. Hypertonic, higher tonicity. It has a higher solute concentration than something else, okay? Hypotonic is the opposite. So if you know anyone who's hypoglycemic, hypo means low. Glycemic refers to the glucose levels in the blood. So hypoglycemic means low blood sugar. Okay, so maybe that'll help you remember it if you know anyone who's hypoglycemic. My sister was hypoglycemic, so that always helps me. So it's the opposite of hypertonic. Hypotonic means a low concentration of solute, okay? Relative to something else. So you could have a glass of pure water, like osmotic, that went through an osmotic filter. It's just the freshest, the purest water you can get. Then you can have just regular tap water, and then you can have salt water. All right, the regular tap water is going to be hypertonic to the reverse osmosis water because it's going to have more solute than the reverse osmosis water. The reverse osmosis has gone through a process that has taken everything out of it. The tap water still has a whole bunch of minerals dissolved in there. Okay, so the tap water is hypertonic, has more solute than the reverse osmosis water. The tap water is hypotonic relative to the salt water. It has less stuff dissolved in it than the salt water. Okay, so a single solution can't just be hypertonic. It depends. It's all relative. It depends on what you're comparing it to. And then isotonic means the two solutions are the same. So you want if you're giving someone an IV, you want the fluid that you're giving them to be isotonic with their red blood cells. And let's talk about why. So here's what happens with red blood cells and plant cells when you put them in different tonicities. So here's your red blood cells, and they're floating around in blood plasma. Blood plasma, that's just the liquid that the red blood cells are floating in. It's water with a bunch of stuff dissolved in it. So normally your blood plasma is isotonic with the red blood cells. So you don't have net water movement in either direction. Same amount of water moves into the cells as moves out. So the cells stay the same size always. Now, if you give someone an IV of pure water, you might wonder, why do you need an isotonic solution in your IV? Why can't you just give someone pure water? That's poison, actually. So if you give someone an IV of pure water, it will cause their red blood cells to burst. Why? Because you're watering down their blood plasma. You're watering down the liquid in the veins through which the red blood cells are floating. So now that blood plasma is more pure water, is more like pure water, and the red blood cells they still have a lot of solutes in them. So the red blood cells are hypertonic to the plasma now because you gave an IV of pure water. Water is going to move down its concentration gradient from where it's purest to where it's least pure. It's going to move into those red blood cells, causing them to lyse. They're going to burst. That's going to kill the patient. Similarly, you may have heard, the saying, water, water everywhere and not a drop to drink. That's like if you're stuck on a deserted island somewhere surrounded by salt water, a saltwater ocean. Don't drink it. You may be thirsty, but do not drink that salt water because it will actually dehydrate you. Salt water in the ocean is hypertonic to your cells. So if you fill your body with all that salt and your blood plasma becomes, the liquid in your blood vein, in your veins and arteries and your blood vessels, if that plasma becomes really, really salty, then your red blood cells will be hypotonic relative to your plasma. That's going to cause the water to move from where it was more pure inside the red blood cells to where it's less pure with all that salt in the plasma. It's going to follow solutes out of the cell, and that's going to cause those red blood cells to shrivel up and die. So that is also bad. Plant cells are different because they have a cell wall, and bacteria also have a cell wall. So you can think of this plant cell diagram to be similar to bacteria. So here's a regular plant cell. This is what happens. Plant cells have the square cell wall. When a plant cell is in an isotonic solution, it's okay. It's okay, but it doesn't prefer it. It would prefer to be in a hypotonic solution. So if you put a plant in pure water, that's good because the water is going to go in and make that plant cell really plump. But the plant cell is not going to burst like the animal cell because of that cell wall. That cell wall is going to it's going to create a barrier. So the cell membrane is going to stretch until it bumps up against the edges of the cell wall, and then it can stretch no further. At this point, the cell will not take in any more water, and the cell wall has protected the cell from overfilling and from bursting. Okay. Bacteria are the same. And this is why, now, if you take away the cell wall from the plant or from the bacteria, their cells are going to burst just like your cells would if they were in a pure water solution. And then plant cells don't, the same thing happens with plant cells as your cells when you dry them out or when you put them in salty water. So if you surround them with salt, the water is going to osmose out from where there's a lot of water to where there's less water. And that's going to cause the cytoplasm to be drained of water. It's going to shrivel up, dry up, and die. Right. I recommend really practicing this, really practicing your vocabulary. You can Google image search hyper and hypotonic solutions and practice identifying which solution is hypertonic between a pair, which is hypotonic, and then also practice identifying which direction the water would move, because I will test you on that. It's also important for understanding why certain antibiotics work, as we've already discussed. So I really do want you to understand diffusion and osmosis, how it affects cells, and also how cells then have to be built to withstand different kinds of osmotic pressure. Bacteria and plants can withstand, their osmotic pressure can get really high, right? They can fill with water, and they won't burst because they have this protective cell wall. That's what this piece of anatomy is for. Animal cells don't do that. So our cells cannot tolerate the same amount of osmotic pressure. Right? So when you're looking at different organisms, especially across the microbial world is very diverse because you have eukaryotes like fungi and protists, and you also have prokaryotes like archaea and bacteria, and they all have a slightly different setup and structure. Some have cell walls, amongst microbes, some have cell walls and some don't. And that's going to be an important piece of information about that microbe and what kinds of environmental conditions it can tolerate. So let's talk about, let me give you a thinking question here. What would happen if a halophile were placed in a low salt environment? So a halophile that means a salt lover. So let's say you have this microbe that's a halophile, that's a salt-loving microbe, and you place it in a very low salt environment. So this is the cell, and it's filled with salts. It's filled with salts so that it's isotonic with the normal high salt environment that it lives in. Let's just say you placed it in pure water here. What's going to happen? What's going to happen if you put it so it was isotonic with its natural environment. In its natural environment, you'd have the same amount of solutes outside as inside. But now you've put it in pure water, what's going to happen? Well, where's there more water? Where's the water more pure? Where's the water more concentrated? Well, now the water's more pure outside the cell, and it's less pure inside. There's a higher water concentration outside, lower water concentration inside. Water is going to move from where water is most highly concentrated to where it is less concentrated. It's going to move into the cell. It's going to follow solutes into the cell, move down the water concentration gradient into the cell, and it's going to make the cell swell until it bursts. So strategies for tolerating high internal osmotic pressure, that's going to be cell wall, right? So there's two, so a strategy for tolerating low osmotic pressure, so a low osmotic pressure in like a salty environment, that's going to be being filled with solutes, right? Because if the outside environment is saltier than the inside environment, water is going to move out, because water follows solute. So you need to at least have the same amount or more solute inside than outside to get the water to move in. But of course, you know, you can only an organism can only tolerate a certain amount of water moving in, so they have to match their environment to some extent. So if an organism lives in a salty environment with very low osmotic pressure that's sucking water out of it, it can adapt to that by being filled with solutes itself so that it's isotonic or even hypertonic relative to its environment so that water stays in or moves in. If an organism is going to tolerate really high osmotic pressure, internal osmotic pressure, because the external environment is more watery, more pure, relative to the inside. The outside is hypotonic relative to the inside. That's what the cell wall is for, because water is always trying to move in. The cell is going to swell and burst if it weren't for this rigid cell wall. So the cell wall, here's gram-positive bacteria. They have a cell wall in red outside of their plasma membrane. Here's a gram-negative bacteria, cell wall in red between the outer membrane and the plasma membrane.